Hello guys and welcome back to Excelsior Academy. So in the previous lecture, we have talked about somatic cell nuclear transfer, right? Now in this topic, in this lecture, today we'll discuss about the pluripotent stem cells. Like what are induced pluripotent stem cells? So we know what do you mean by stem cells, right? Now here pluripotent मतलब क्या हुआ और induced pluripotent cells मतलब क्या हुआ? So we'll discuss this in this topic today. Okay, so first let's understand what is the meaning of pluripotent. Okay, so pluripotent over here, it means that the cell is able to self-renew. So we know that this is possible in the case of the stem cells. So there is one stem cell is there, it can self-renew and it will give another cell as for the differentiating, right? So this we know, this is a beautiful property of any stem cells. Now in the case of the pluripotent stem cells, of course it can self-renew and it will develop into three types of the three groups which can give rise to the differentiated cells. Now what are these three? So those three are nothing but your ectoderm, first group, mesoderm, and then it is your endoderm, right? So we know that ectoderm kya karega, it will give rise to your skin and the nervous system. Mesoderm is forming the gastrointestinal and the respiratory tracts, right? Sorry, that is your endoderm. Endoderm is forming the your gastrointestinal and the respiratory tracts. It will also form your endocrinal glands also, right? Whereas in the case of the ecto, it is the outer. So this pluripotent stem cells will self-renew and give rise to these three groups of ectoderm, mesoderm, and endoderm. Now, when you say induce, it means you're forcing them to become a pluripotent. That is, we will first de-differentiate the cell. It will go and it will become pluripotent. So we'll see how this induction is being done. So given both the technical and ethical, given both the technical and ethical difficulties in the derivation of the embryonic stem cells by the somatic cell nuclear transfer, a major advance in the field came from the studies demonstrating that adult somatic cells can be directly converted to pluripotent stem cells in the culture. So what we are saying over here is that you have got your somatic cell here. Okay. So somatic cell means it is already the differentiated cell. Now you're going back, you're inducing them, by giving the proper transcription factor or the proteins which is required to convert them. That is, यहाँ पे हम इसको de-differentiate कर रहे हैं to become your pluripotent stem cells. Okay, now this circumvents the need for derivation of embryos, provides a direct mechanism for converting somatic cells into the stem cells, which is like embryonic stem cells, and then they have the potential to develop into the tissues of an organism. So once you get the pluripotent stem cell, then you can give the required transcription factor so that they can differentiate into the required cells and form the tissue. Okay, I hope you're getting till here. Now, one second. Okay, so given the conversion or the reprogramming of somatic cells to the pluripotent stem cells, it was first reported by the Kazuyoshi Takahashi and Shinya Yamanaka in 2006. नाम से ही पता चल रहा है Japanese है. Okay, so they found that these mouse fibroblasts could be reprogrammed to the cells forming the embryonic stem cells. Okay, now this is by the action of only 4 transcription factor, which was introduced by the retroviral gene transfer. So what they have done is that they have got the somatic cell यहाँ पे, somatic cell से इसको इन्होंने pluripotent stem cells किया है, and for that they have used the retroviral method of the gene transfer for the transcription factors. Those are 4 transcription factors over here. Okay, now subsequent studies have shown that induced pluripotent stem cells like embryonic stem cells are capable of differentiating into all types of when introduced into early mouse embryos. So iske baad, once you get the pluripotent stem cell, then you can differentiate into the respective cells. Okay, so here you have got the adult mouse fibroblast cell is there, and you have infected this cell with the help of the retroviruses, which carries the genes for OCT4, SOX2, KLF4, and the CMIs. Now, these transcription factor will convert the somatic or the fibroblast cell back to the pluripotent stem cells. Now it is being induced. Why? Because of the presence of these 4 transcription factor. So guys, this is important. Please, 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 isko yaad rakho. These are the 4 transcription factors which is required to convert any somatic cell into the induced pluripotent stem cells. OCT4, SOX2, KLF4 and the c-MYC. Okay. Now, retroviral vectors. तो कैसे हमने किया है? So what is the technique behind that? So this vector, it will consist of the retroviral sequences which is cloned in a plasmid that can be propagated in the E. coli. So here you have got the plasmid DNA. यहाँ पे रेट्रोवायरल सीक्वेंस है, हियर द क्लोनिंग साइट इज देयर, राइट. तो ये जो क्लोनिंग साइट रहता है, यहाँ पे आप अपना जीन, जो रिक्वायर्ड जीन रहता है या इंसर्ट डीएनए है, दैट यू आर गोइंग टू इंसर्ट ओवर हियर, राइट. नाउ यू आर गोइंग टू आइसोलेट दिस प्लास्मिड, इंसर्ट योर रिक्वायर्ड डीएनए. Now this introduced DNA, it is then transfected into the mammalian cells. Transfection. So transfection यहाँ पे क्या हो रहा है? So you are going to take the virus which carries the gene of interest in the plasmid, which is present in the virus, and that virus is going to infect the mammalian cells. Now what will happen is that these few mammalian cells will take up the DNA and produce the recombinant virus particles. So what is happening now? So these recombinant retrovirus particles carry the inserted DNA. यहाँ पे हमें बहुत सारे copies मिल गए, ठीक है? Now you take this and infect in the new cell जहाँ पे हमें ये चीज चाहिए. So what is happening then? This particular retrovirus is infecting the cell. For example, let's say this is the fibroblast. अब वो infect करेगा, so what will happen? ये जो gene carry कर रहा है आपका retrovirus, that will get integrated into the host genome. Now, once it is integrated into the host genome, what will happen? That will be translated, that will be transcribed and translated, and hence you get the protein. And we know that the transcription factors are the protein. So then these transcription factors will be expressed. It means on the expression of these transcription factor, now this particular fibroblast cell has got converted into the pluripotent stem cells. Okay. So let's see what are the steps given here. The vector, it consists of the retroviral sequences which is cloned in a plasmid that can be propagated in the E. coli. Now, the foreign DNA is inserted into the viral sequences and recombinant plasmids are isolated in the bacteria. Animal cells in culture are then transfected with the recombinant DNA. Now, this DNA is taken up by the small fraction of the cells which produce recombinant retroviruses carrying the inserted DNA. These recombinant retroviruses can be used to efficiently infect the new cells, where the viral genome carrying the inserted gene integrates into the chromosomal DNA as a provirus. Okay? Now, further research has also demonstrated that adult human fibroblasts can be reprogrammed to the pluripotency. So now it is possible to convert your skin cells, which is taken from a patient, directly into a stem cells, and stem cells को हम induce करेंगे so that the differentiated cell बन सकता है, and that will be helping in the transplantation therapy. तो की फैक्टर क्या है यहाँ पे कि वो कौन से ट्रांसक्रिप्शन फैक्टर्स हैं विच वी नीड. नाउ गाइस, ये बोलने में जितना आसान है उतना ही डिफिकल्ट है, बिकॉज यू नो दैट क्लोनिंग में कितने प्रॉब्लम्स आते हैं. लाइक यू नो समटाइम्स दी डाइजेशन इज नॉट प्रॉब्लम, समटाइम्स द लाइगेशन इज नॉट प्रॉब्लम. सो बिकॉज ऑफ दिस द एफिशिएंसी ऑफ थेरेपेटिक क्लोनिंग जो है वो थोड़ी सी रिड्यूस हो जाती है. ओके. Now, pluripotency transcription program. So these are the core transcription factors which is required OCT4, SOX2 and the nano which maintain the pluripotent stage. So these factors form the positive autoregulatory loops as well as activate genes which will maintain the pluripotency and repress the genes that induce the differentiation. So now you know, जब तक ये एक दूसरे को induce करते रहेंगे, these 3 transcription factors will maintain the pluripotency of that particular cell. Okay. Now, these factors form an autoregulatory loop in which it means they are going to act together. That is, एक दूसरे को वो positively regulate करेंगे. Okay. Now, this positive autoregulation at the core of this transcription program maintains the pluripotency of the cell while allowing the cells to undergo differentiation in response to appropriate signals. Now, apart from these 3 signals, if you get another transcription factor, okay, let's say the CMIs, okay, then it is given. Now, this will result in the differentiation. So, it is noteworthy that autoregulatory maintenance of pluripotency has allowed investigators to overcome major obstacle to the potential therapeutic use of induced pluripotent cells. For example, c-MYC. Now these are they act actually the oncogenes. अब भी क्या है, during this process if this gets mutated, then that particular cell instead of becoming stem cell it will become what, the cancer cell. ठीक है, अब आप उस चीज को यूज करोगे, so what will happen, instead of having the therapeutic effect you are actually introducing cancer in the patient. So that is what we need to take care when we are doing this retroviral transfection of the cells. Okay. So, however, methods have now been developed to establish the pluripotent stem cells by the transient expression of the reprogramming factors without the use of the viral genomes, because the chances of mutations are high when you're using the retroviral transfection. Now, once the recipient somatic cell is reprogrammed to the pluripotent state, autoregulation maintains pluripotency without the need for continued expression of the factors that initially induce the reprogramming. Okay. Induced pluripotent stem cells generated by transient expression of reprogramming factors thus provide a ready source for the pluripotent stem cells for the transplantation therapy. So now we can say that then there is some hope that we can have the therapeutic cloning. Okay, now this is a different topic. Like, what do you mean by transdifferentiation? Now, the success in reprogramming somatic cells to induce pluripotent stem cells by the different methods. Okay, that is transdifferentiation. यहाँ पे हम लोग retroviral method use नहीं कर रहे हैं, ठीक है? We are using different method. That is, for example, let's say the transcription factor MyoD was sufficient to induce differentiation of fibroblasts into the muscle cells. So here we are not making your somatic cell, somatic cell to your pluripotent stem cells. What we are doing here is that we are making one cell to another cell. For example, differentiating fibroblast directly to the muscle cell. So this is transdifferentiation. यानी कि एक पर्टिकुलर सेल से आप दूसरे सेल में आप उसको डिफरेंशिएट कर रहे हो. For example, you have taken the fibroblast. Now, if you activate the transcription factors like GATA4, MEF2, CTBX5, then this fibroblast will get converted to the heart muscle cells. If you activate these transcription factors, ASE1B, M2, MYT1L, then it will get converted into the nerve cells. So this is the meaning of transdifferentiation. So इसमें थोड़ा सा success मिला है. Now you can also go for differentiation of the blood cells, which can be reprogrammed either to the blood cell type to the hematopoietic stem cells. Again, you're providing another therapy like hematopoietic stem cell transplantation possible है यहाँ पे, ठीक है? For example, transdifferentiation thus provides a direct route to generate desired types of differentiated cell populations, bypassing the need for becoming the pluripotent stem cells. यानी कि हमें अभी pluripotent stem cells बनाने की जरूरत नहीं है, एक सेल से हम दूसरे टाइप ऑफ सेल में हम जा सकते हैं directly. So for example, let's say that you have a cancer patient. Okay, so we are doing hematopoietic stem cell transplantation. So let's say that we have a cancer patient over here जिसको ट्यूमर है. अब आप क्या कर रहे हो, हाई डोज ऑफ कीमोथेरेपी दे रहे हो, जिसकी वजह से क्या होगा, डेफिनेटली the drugs are going to kill the tumor cells, but along with that it is going to kill the hematopoietic system also. So now this person does not have any immune cells also, because आपका पूरा hematopoietic system चला गया है. Okay, so this person is very susceptible to even one bacteria, because that one bacteria can kill a person. So they will be kept in a chamber where the air which is being pumped over there is going to be bacteria free also. Okay, now this person will undergo or receive hematopoietic stem cell plant. Again, the restoration of the hematopoietic system is happening. So हमने यहाँ पे क्या किया? What we are saying is that पहले आप सारे kill कर दो, then instead of adding these stem cells, what you can do is you can just take one cell and differentiate them into the hematopoietic stem cells, and that can be added. Okay, so you can take any cell. आप कोई भी somatic epithelial cell ले सकते हो, उसको आप differentiate कर सकते हो into this hematopoietic system, hematopoietic stem cells, and that can be transplanted. And यहाँ पे rejection का problem नहीं आएगा, because it is being taken from the patient only. And वही आपका receiver है, वही आपका donor है. So the chances of having the rejection is going to be very, very, very less. Okay, so this is how this also we have discussed before, like how the hematopoietic stem cell transplantation can be done. And this is another example where you don't have to have the stem cells. You can just directly go also. Okay, so that's all guys. I hope this was interesting. So the takeaway message over here is that you know what is pluripotent stem cells. Now we also have different option. The alternate option is that not going back and making the pluripotent stem cells. What we can do is, see guys, ये बहुत simple है to understand. So first you, let's say that you had the fibroblast, ठीक है, इसको आपने pluripotent stem cells में convert किया, and then this is being differentiated into muscle cell, or we can say nerve cell. Now what we are saying is that with the help of the trans differentiation strategy, fibroblasts can be directly converted into the muscle cells if you have got the proper activations of the transcription factors as mentioned before. So अभी ये जो स्टेप है आपका स्टेम सेल्स बनाने का, इसकी हमें जरूरत ही नहीं है. डायरेक्टली फ्रॉम वन पर्टिकुलर टाइप ऑफ सेल यू कैन गो टू अनदर टाइप ऑफ सेल्स, and this will help in the transplantation therapy. Okay, so I think in future if you're interested guys, you can read more about what is trans differentiations. और अगर कुछ डाउट आ रहा है, you can please ping me back and you can ask. Okay, so that's all guys, thank you.